So our next talk is going to be Griffin, and they're going to be talking about metabolic profiling in Shenandoah. That's forward, backwards, and that's kind of like a little pointer thing.
Okay, thanks so much. Is the microphone here too?
It's right here. You can use this one, or you can use that one.
Okay. There we go. All right. Hello, everyone. I'm going to try and keep this to 50 minutes or less. We'll see how I do. But yeah, my name is Griffin Erich. I'm, as of a few weeks ago, a second-year PhD student at Cornell. I'm mainly doing research on apple fruit and the fruit cuticle, but I've always had a passion to do research on pawpaw, so I've been really fortunate that one of my committee members, Joseph El Shafi, has a few grants dedicated to studying pawpaw fruit, and really excited to present the results of this Metabolomics project with y'all. So, yeah, just a little bit of background about myself. I first got into pawpaws a few years ago. One quite literally hit me in the head when I was walking around campus, so I feel like that was kind of forthcoming. But ever since then, I've really enjoyed doing research on these pawpaw fruits. Maybe some of y'all have seen my poster outside yesterday. I did a little bit of work with 1-MCP applications and trying to elongate the shelf life of pawpaw. Unfortunately, we didn't really see the results we expected with that. I understand the climacteric peak of pawpaw is very short, and also very specifically, there's a very short window after the harvest time, so we might try and repeat that experiment again in future years. But the current work that we're doing at Cornell is focused on non-destructive ways to determine pawpaw maturity using near-infrared spectroscopy. We're trying to determine the Brix levels and dry matter levels non-destructively. And then also the work I'm going to be presenting here today on the fruit metabolome of Shenandoah. So I guess I always start my pawpaw presentations with this by saying, with a show of hands, who here knows what a pawpaw is? But I think if I did that here, everyone's hand would automatically be raised. So yeah, I'll skip that one for now. But yeah, so on the left here we have a pawpaw tree that's in commercial production. As you see, it has the pyramidal shape, right? The leaves kind of face downwards, and the tree typically grows between, let's say, 15 and 20 feet tall, maybe 30 feet in some instances. I think this is a really nicely maintained orchard, and they have a central leader type figure to the tree. And on the right side, we have a pawpaw cultivar. It's got a really nice orange yellow color with a nice custard-like texture. We've got a few seeds in there. I think the New York Times did a really nice photo here. So I got this data off of the Agricultural Marketing Research Center, but typically pawpaw is sold from about $4.50 to $5 a pound. Now, I understand that can vary significantly in terms of geography and location, right? I mean, there are some places, I think there was a presentation earlier that mentioned they were selling pawpaws for up to $10 a pound, and I've heard in New York City they can actually go up to like $20 a pound in some instances. So, in luxury markets, you can see the price of pawpaw elevated in a lot of cases. But needless to say, the pawpaw does have a very important role as a specialty crop and a pretty high economic return. So another thing I would like to point out, that pawpaw is quite nutritious. I think this is there's a few studies now from Neil Peterson and also Robert Brannon, who's here, talking about the role of or comparing pawpaw's nutritional profile to the same serving size of apples, orange, and bananas, and pawpaw actually outperforms most of these other common horticultural commodities in terms of protein, most minerals except for potassium, essential amino acids, and also niacin. So it's just a good way to show that pawpaw is quite nutritious and has a big nutritional value. And then in terms of medicinal purposes, I guess I'll kind of breeze through this pretty quickly, but we've heard about the acetogenins throughout the day now. They do some work in disabling the complex 1 of the ETC during cellular respiration. So they have basically the ability to lead to cell death, and this has been used in anti-cancer research from Memorial Sloan Kettering Hospital. I think it's been pretty interesting that there is this preliminary research so far, and hopefully this can kind of emerge as also a crop with some medicinal purposes as well. And so this all connects. We have the nutritional profile of pawpaws, we have the medicinal profile of pawpaws. It all relates back to the fruit metabolome, which is essentially the collection of these small molecular weight compounds that we call metabolites. And the metabolome is responsible for determining the taste of the pawpaw. It's responsible for determining the medicinal properties of the pawpaw, and something that really has not been well tapped into as of yet. But essentially, Joseph and I, we were very interested in understanding how this differs between both the peel and the flesh of the fruit, and also throughout the harvest season. So at 3 different points. So yeah, that kind of ties into our research questions, right? How is the fruit metabolome altered in Asimina, Triloba, Shenandoah across the 3 different harvest times? And also, how does the fruit metabolome differ between the fruit peel and the fruit flesh? So to investigate these questions, we designed an experiment where we harvested fruit from, if y'all are familiar with Jim Davis's orchard in Maryland, last year we harvested on 3 different days, September 8th to be the early harvest, September 15th to be the middle of the road harvest, and September 22nd to represent the late harvest of all of Shenandoah. And to analyze the metabolome of both the flesh and the peel, we used a cork borer of the same size, and we immediately flash froze the samples in liquid nitrogen, so around -80. And then we shipped them out to Metabolomic Center out in Boston, Medaware Biosciences, and they did a bunch of work with this technology right here, ultra-performance chromatography, ultra-performance liquid chromatography, tandem mass spectrometry, say that 10 times fast. I like to say UPLC, MSMS. But essentially, this is a really cool piece of technology that's spread across all of biology, right? We have the liquid chromatography phase, where these compounds are separated based on either their polarity, their size, their charge. Then they're sprayed with this electrospray ionization thing. And basically, after that point, they're analyzed on their mass-to-charge ratio. So each of these compounds has a unique molecular weight. And comparing this to a known library of compounds, we can identify the vast array of metabolites that are present in pawpaw. So actually, using this approach, we were able to identify over 3,000 metabolites between the peel and the flesh of the pawpaw fruit Shenandoah. So going into our results, this is just a broad profile of what we've identified in terms of compounds by their specific class. So we've seen that I think the most abundant one would be the flavonoids, so like secondary metabolites. We've seen a big profile of alkaloids. This one is primarily found in the pawpaw peel as defense compounds. And we've also seen some other specialized metabolites as well, like terpenoids. We've seen some steroids, lipids. And it's really given us a pretty ubiquitous, or I should say a pretty widespread look at the entire metabolome and all these different compounds that have been within pawpaw fruit. This is a PCA plot trying to determine how the different groups and the variants of the compounds differ between both the tissue type and also the harvest period. So I think the main thing to take away from the PCA plot here is that we see the samples from both between the same tissue type and then also the same harvest time are often grouped close together. Like for green, for example, for the flesh in harvest 1, the samples are pretty close together there in the bottom left-hand part of the graph. And this seems to be the case, right? So within each harvest, the samples are relatively close together. But if you look again, there's a large difference in the way that the samples are characterized between the peel and the flesh, right? So really to the left-hand side of the graph, we see most of the dots representing the peel. And on the right-hand side, the dots representing the flesh. On the right-hand side of the graph, we see many of the points representing the peel. So there is a big difference, or a larger variance, in the way that the peel metabolome is contrasted with the flesh metabolome. So this graph here is maybe kind of also trying to show the same thing, right? Where on the x-axis we have the log fold change, or this is essentially the absolute value of the difference between the metabolites in the peel and the flesh. And on the y-axis, we have the class of compounds we're looking at. So starting with alkaloids, going all the way down to terpenoids. It's also characterized by color in the class in the key there. And the variable importance and projection. So that just basically means we're taking the certain metabolites, which is represented by each circle, that were above 1.05 in terms of importance. So you don't see 3,000 dots on here. That would be a little chaotic to try and depict to everyone. So we're just showing the ones that are most important, which in each class. And as you can see, we do see that there are certain compounds within the alkaloids, within terpenoids, within the other category as well, within lipids, that are pretty big in terms of differentially expression. There's a very large difference in the way they're differentially expressed between the peel and the flesh, which basically further confirms that the pawpaw peel metabolome has a bit of a different profile compared to the flesh metabolome. This was on Harvest 1, and the same trends are quite apparent too in Harvest 2. See the same trend with alkaloids, and the same trends are quite apparent in Harvest 3, this time again with the terpenoids. Essentially all these graphs are just showing that the peel and the flesh metabolome are quite different. So some conclusions we can draw from this study. The metabolome of the pawpaw peel and the flesh are vastly different in each harvest, as these graphs were just depicting. The metabolome of the fruit and peel respectively change with harvest time. So that's another difference we noticed as well, is the metabolome of the peel and of the flesh, they tend to change as you progress in terms of the pawpaw maturity. And then third, we didn't notice too much variation between the samples at each harvest time in terms of the peel and the flesh metabolome. So the peel metabolome and the flesh metabolome was quite conserved among the samples between each harvest time. And then some of the future research we hope to do based off of the metabolomics data. One would be to identify some compounds that serve as biomarkers for quality traits in pawpaw breeding efforts, right? So again, these contribute to the way that we perceive the taste of pawpaws and possibly also their storage life as well. So we hope this metabolomic data can be factored into breeding decisions. Second, we hope to use these compounds, try and unveil any additional purposes they may have for medicinal importance. I know there's been a lot of pharmacology work done on pawpaws, but of course, this study, I hope to look at the data a little bit more, but I believe that there are some compounds that are newly identified and have not been revealed to be in the pawpaw flesh or peel in the past. And then third, we hope to use this data to establish a pawpaw metabolite database. This is something I'm kind of hoping to work on over the course of my PhD on the side, and we'll see how well that goes, but I think that would be really important to have there and kind of establish a library just for pawpaw fruit. And I'd just like to thank my funding sources from the TSF, the Tourist Sustainability Foundation, Cornell Cooperative Extension, and one of my committee members, Dr. Yosef El Shafi, and of course Jim Davis and the Deep Run Pawpaw Orchard for making this project possible. Any questions?
Do we have time for a question or two? So, questions? They're bringing the mic over.
Similar research on the fruit as it goes from in the ripening process to over-ripening. This is just the same stage of fruit at different times of picking, right?
Do you mean like during storage or? Oh, I see what you're saying.
This was all different days in the month of September.
Yes.
But I would be very interested to know what you found if you track fruit after it's been picked as the days go by for that particular fruit. So the aging process.
Yeah, that would be interesting to do as well. We haven't had the chance to do it yet, but I think understanding the post-harvest metabolome, if you'd like to call it that, would be another advancement in trying to understand how storage is affecting the way that these compounds are expressed and or produced, I should say, and how that's contributing to things like the flavor profile as well. Thank you so much. Appreciate it.